Thanks, Ivar, and thanks for inviting me to this event. I added the missing oil at the title in the program. It was so I tried. Well, it's oil crops that we mainly are working with, and I'm presenting results from some couple of years' work at Alnarp, which is in the south of Sweden. So belonging to the SLU, and it's well, it deals with this, so to say, dilemma that we have in the GM, that we have kind of a building up of a lot of opportunities that, for reasons, cannot reach the market. So what I'm presenting is also, I'm at the, since a year I'm also ahead of the department, so I'm presenting a lot of the results from my colleagues. Well, starting with this, a little bit of introduction. This is like the sector we are dealing with and approaching from the plant side. The fact that we have a society that's very based and depending on the fossil fuel. 90% of the fossil fuel goes into fuel, 10% goes into petrochemical as a feed source. Out comes all of these necessary products that we have in our society. So the idea is, how can we deal with this? How can we replace the fossil fuel? We believe that plant oil can have some importance here. Also from the fact that fossil fuel, basically carbon chain, is very similar to the plant oil molecular structure. There are some differences that makes some importance with the plant oil compared to the fossil oil. But in general, there is very much in similar. Knowing that fossil oil mainly is derived from plankton or algae over millions of years, and then so to say made into what's now is fossil oil. So the chemical structure is similar, and that is also in importance. Looking at the plant oil production, this is some figures, 2012, 160 million metric tons. That's what the total production of plant oil. You can see that it's split up on what is it now, 10, 11 something sources. It's interesting to note that the plant oil production in the world are based on basically four crops: palm, soybean, rapeseed, and sunflower. It's making up more than 80% of all the production. So it's a very narrow production base, which is also something that one should reflect on when you come up and say, let's replace fossil oil with plant oil. Because palm, there is diseases that can wipe out the plant palm oil production, and then we're losing almost a third of the production. So that's just a comment in that. Another thing is that if we want to replace fossil oil as a feedstock in the chemical industry with plant oil, we need to observe that today 80% of the plant oil is used as food. So that is also a challenge. We can have two options. We can think about the fossil fuel and the amount that goes and used as fuel, replace that with the plant oil. Consider taking all of the plant oil production today, which is 160 roughly a million tons. That makes a very narrow, very small slice of the total amount of oil that would be needed if we are thinking about replacing the fuel application. Instead of looking at the 10% of the fossil oil that is used as a feedstock in the chemical industry, that's roughly 400 million ton. Using plant oil can replace a much bigger slice of that. And we believe then, from our field, that it would be makes more sense to use the plant oil to replace the fossil oil that goes into the chemical industry as a feedstock. That is more relevant. It's more, how to say, well, it's a better use of the resource than just use it for fuel, burning the thing. And we also believe that replacing the fossil oil going into fuel would need different other kind of techniques, solar, wind, whatever you can think about. And instead, feedstock is what we believe we should use the food oil for. Sorry, the plant oil. Coming into what should be producing the food, the plant oil. Well, we have oil crops, but as you saw, we're using, of course, the plant oil for food and for industrial oil. And we think there is a sort of a consideration. We need to have a risk mitigation here. We don't want to mix these two things. So looking at the sources, you can think about it as multi-purpose oils. That is then plant oils that can be used either for food or for feedstocks. And a good example of that is oleic acid. 18-1. It's an 18 carbon long fatty acid with one double bond. It's very, very common oil quality. You find it in food oil. It's excellent to cook, to use as a food cooking oil. It's also excellent to make biofuel, biodiesel, if you want to go that way. But it's also an interesting source to use as a feedstock, because it's as a double bond roughly at the middle of the carbon chain that can be cut and then get two other structure that is interesting for the industry. So multi-purpose oil can be produced by oil crops and not thinking too much about risks. Just use it for food or for feedstock, because that's what it can be used for. Then we have what we can call technical oil qualities. These are oils that contains fatty acids with, as we said, functional groups. And you have these functional groups, epoxy, hydroxy, conjugated, so on. These are fatty acids that has a molecular structure that doesn't necessarily make them poison. But some of them doesn't, you don't feel good if you eat them. You may get harm from some of them. But anyway, it's structures, molecules that we want to keep separated from the food chain. So therefore, technical oil crops should then be preferably produced by crops that's not linked to the food chain. When we're looking then at what kind of oil crops should we consider for producing these technical oils, well, we need to have then some requirements. The product should be high quality, should be low price. Use of existing production infrastructure is also of relevance. They should not have an environmental impact. These should be dedicated non-food oil crops producing technical oils. Identity preservation and low risk of outcrossing that part of the game to try to keep them separated from the food chain. And we should also have tools available which we can breed and develop these crops. So these are reasons, these are important considerations when we're thinking about, okay, what crops should be producing the technical oils. The results I'm presenting so to say, from where we have tried different types of techniques. A lot of it's GM. And we have a couple of groups that are working with this area of looking into how can we use plant material as biomaterial, as replacement for fossil oil. Icon was a big public-funded EU project that was ended last year, coordinated by Sten Stumne at our campus in Alnarp. And the idea with this was that, well, this is one of the oil qualities that this project worked on a lot. Jojoba is a sort of say well-known plant, and it produces a special oil that has very excellent lubrication properties. It's wax esters. These wax esters has a special molecular structure. You have a fatty acid that is linked or connected with a fatty alcohol. And if you compare that with the common oils that you would find in all of our normal oil crops, you would see that there is a structural difference here. You have three fatty acids here, which looks different. So it has a different molecular structure. But with the wax esters, this excellent lubrication properties makes it excellent for making products that needs these properties. Problem is that it's very expensive to grow, so the end product is very expensive. So that means that it can mainly carry the cost in high-end products like cosmetics. And that's not because it can't be used for making engine and machine lubricants. It would be excellent there, but it's too costly. So the idea one of the ideas with this product was to see, can we make this production happen in a common oil crop that could be produced cheap on big areas. So production of biolubricants in the form of wax ester in industrial oil crops using plant biotechnology, that was the essence of this project. And the choice of the oil crops then was picked using these requirements that I showed earlier. So it should be then that they can't easily cross with anything else, and it should be able to grow, it should be able to be developed using molecular techniques and so on. So these were the three crops that were identified as the platforms: Cramba abyssinica, brassica carinata, camelina sativa. None of those are oil crops that have been grown a lot, but they have been grown. So they were not totally unknown for people. Another area was that, well, when you had squeezed out the oil, extracted the oil from an oil crop, you have some left, which is called a feed cake, basically. It's very high in protein. Normally, when you take rapeseed, for example, that feed cake is turned into feed and used as that. But in this project where we are looking at technical oil qualities, the idea was that we didn't want to mix this with food or feed. So the idea was then to look, can we use it for something else, for biomaterial? So together with one of the partners, company in Vencia, and a group at the university, which is represented by Eva here, they were looking at, can we use that as a base for producing plastics from it, in this case containers, for example. And the sort of say the technical engineering of the plants were in a way pretty straightforward. This is showing that in these three different oil crops, the same kind of engineering, adding of additional genes critical for changing the pathway or changing the production of the oil was done. And so the idea of this, not trying to be too technical here, but basically it's like the fatty acid is produced in the chloroplast and then exported out from the chloroplast in the form of 18 carbon long fatty acids. And then this 18 carbon fatty acid is elongated, added up to 20 and further into 22 carbon long. So now we have a 22 carbon long carbon chain. Then that is converted into an alcohol. Okay, now we have a 22 carbon long alcohol. We also have 22 carbon long fatty acid, 2 types of carbon chains. And then this was done by 2 enzyme here that was introduced. And then a third enzyme over here takes the fatty acid and the alcohol and put them together like this, if you remember the structure, making it into a wax ester. And the wax ester quality here is the same that we can find in the jojoba. So that was pretty much, this was done then by transgenically introducing these 3 genes into these crops. At the same time, in this project, there was, you can say, a hunt of other types of these enzymes. Around in nature. And you can see that it's pretty wide in where they were looking. They were looking for other types of fatty acid reductase, the one that makes the alcohol, and enzymes that put them together into a wax ester. This one, for example, they were looking for these genes at the back of the owl, you know, because they have resins, I think. Oh, yeah. They rub the beak and then they rub the feathers. This is wax esters. Very special, very unique kind of wax esters with excellent properties. And the same with all of these other creatures. They have unique wax esters produced by these genes. And so why is it important? Well, the thing is that this molecular structure, the wax ester structure, a fatty acid and a fatty alcohol, you can vary that structure a lot, changing the length of the carbon chain of the alcohol, changing the length of the fatty acid carbon chain, adding different types of active groups here, and all of these changes will make specific wax esters that have specific properties. So if you know the properties needed to make a specific product, lubricants or whatever, having this sort of toolbox with a lot of different specific enzymes, specific genes that make these enzymes, you would be able to modify and design specific wax esters qualities. So, out of this, after a couple of years, the result was that this jojoba type quality of oil, they were able to show that they could produce that in all of the 3 crops. So if you see here, you have in these are carinata. These dots over here show that you have wax ester produced, as well as the common oil. From the beginning in carinata, in all of these 3 crops, they all produce just ordinary oil, which is triacylglycerol, which is this dot here. The transgenic then produce these extra wax esters. Down here, you can see an example of how they could show that in camelina, in this specific camelina, they also produce wax ester. Because in the non-transgenic camelina, you have the oil there, you put it in the freezer, and it still it comes into one phase here. While in the transgenic that contains some wax ester, which has a different kind of quality, the wax esters solidify and showing that it's there. Then another stream in this project was to look into another type of quality, and this is Urusic acid that is present normally in cranberry, which is one of the 3 oil crops. So cranberry has actually been grown specifically because it contains already from the beginning roughly 55 to 60% of the fatty acids made up of erucic acid. So cranberry has been grown because it is rich in erucic acid. 55 to 60% still needs to have some, purification when you extract it. So the higher concentration of the specific fatty acid you have in the oil, the better it is. So this part of the project was just to see how can we increase the percentage of this fatty acid. That was also done in the same way, transferring into the plant-specific enzymes. And the key for success in this specific product is to look at the common oil molecule, which is 3 fatty acids like this. In cranberry, the normal cranberry, you don't have on this position any erucic acid because cranberry lacks the enzyme that can put the erucic acid on the second position. This enzyme, which was taken from another plant, can do that. So at the present, they have then cranberry that have up to 77% erucic acid. So this year and 2012, there was a field trial done on these 2 oil qualities, the wax esters and the erucic acid. 20% wax esters and 73% erucic acid. And specifically the erucic acid, that cranberry is today at the level or at the point where you basically could grow it on the land for a farmer, and would probably be profitable. So Ivor could probably grow it and get money out of it. There's buyer from it if we don't. Well, if we had different types of regulation here in it was considered to be a very good project from the EU perspective, even though in this environment of criticism. At the same time, there are other groups that is connected to this project, to this network, that are growing and working with other oil qualities that are also interesting as feedstock in the industry. So there's 2 problems besides the problem with the GM problem controversy, and that is, as I was saying from the beginning, we have too little plant oil today to really make a difference in doing this replacement of the fossil oil. So another field that we are also looking into is how can we increase the amount of oil production. And that is a completely different story. But I just make it short that how we are looking at this is that we think that in the future we need to think about oil crops in a different way than today. Today we're thinking rapeseed, sunflower, palm oil. We're thinking in the future we need to use oil production that are making, taking advantage of other organs, other parts of the plant, roots, stem, and leaves. And we're also looking at using the molecular techniques and the tools we have today to remodify the carbon allocation into different, in this case, going into, these are models, you can say, wheat and maize, but I mean, they have the strategy to accumulate energy as starch. but they all are using the same starting material, which is the sugar from the photosynthesis. So what we're looking into this, so basically look at how can we channel the sugar into oil instead of starch. We're looking into sugar beet to channel that energy that goes into sugar instead of going into oil. So, and that is a challenge, but we have some interesting results going there. So, thank you.